In this short video, I'm going to show you how to use what is known as a multiple sequence alignment, or MSA, in order to figure out what parts of a group of, in this case, protein sequences are best to use for constructing a phylogenetic analysis to understand evolutionary relationships. Now, multiple sequence alignment is in some ways similar to BLAST, which you used before, but it's also in some ways quite different. They're both alignments, which means I'm trying to see the optimal way in which they share certain parts of their sequence in places where they don't. But unlike BLAST, which only shows pairwise alignments, a multiple sequence alignment uses two or more sequences at a time. And multiple sequence alignments are global alignments, which means they use the entire sequence to try to match up, whereas BLAST is what's known as a local alignment. It only finds the best sort of subsequence that matches. Now, it may be the whole sequence, but it doesn't have to be in a BLAST search. And also, unlike BLAST, a multiple sequence alignment is not searching a database. Rather, it uses a small set of sequences that I, as a user, have already defined as being of interest to me. Okay, so we've been assigned our protein sequences from our bird lice ecotypes. We've used one of those sequences to go find a human louse outgroup, and now we want to figure out to what extent do these proteins really share a lot of sequence conservation that would allow us to believe they truly are homologs and therefore can be used in a phylogenetic analysis. So I'm going to go ahead and copy these sequences. And we're going to go, as shown here at the bottom of this document, we're going to go to a tool called Muscle, which is one multiple sequence alignment tool that is quite popular these days. And here is its URL. Okay, so we're going to go to Muscle, and it tells us a little bit about it, and we're going to go ahead and paste in our sequences. Okay. Now, one thing I should say to you here is that these sequences are in a particular format known as FASTA, or FASTA format. And what that means is we see this little greater than symbol, or caret, and then some sort of name of our sequence, and then the sequence. And that is a common way that a lot of bioinformatics tools knows where one sequence ends and another sequence begins. Now, because we're going to be shown an output where every one of these sequences is going to be on a given line, it helps to have this name be a little bit shorter. And quite honestly, a bunch of letters and numbers really aren't going to tell me much. So I'm going to just do a little bit of cleanup here, in which I'm going to reduce each of these names just to the genus name of the organism. And in that way, I can make this a lot cleaner in the way it looks in terms of output. And it just takes a second. All right, and once you do this, you might want to copy it, and that way you can hold on to that cleaned up sequence later on. Almost finished here. All right, and I think I'll leave the name here on the top one just so I have a name of what gene I'm looking at here. So we're going to use the defaults of Muscle for the moment, and we're going to go ahead and submit that. And literally what it's doing is trying to find the best ways in which these set of proteins overlap in terms of conserved sequences. And depending on whether or not we had whole protein sequences or not, we may or may not see the whole sequence line up. So here we go. It's quite fast because we're only comparing, in this case, 10 sequences, unlike BLAST, where it was remember, it was looking through millions of different proteins. So here's the best alignment you can find, and right away you can tell it's done actually a really good job. Look at this conservation here. Now, where you see dashes, that means a part of a protein isn't present, There's nothing to line up there, so it puts a dash in because it has to go from the start to the finish. So here's the largest sequence, and these guys don't have this out here, right? And some of these sequences are obviously incomplete. But if we look down here, we can find areas denoted by these stars where every sequence is exactly the same at that position. And so in fact, notice along this sequence here, as we just scroll through, this protein is highly conserved, even across a wide range of these different lice. And so these are truly homologous proteins. Okay, and so we can use that in a phylogenetic analysis. So we come back here. It looks like right in here is where we can first start to say that all the sequences have something we could say this is the start of a conserved region. And so we can, start there and try to construct the subsequence we're going to use for a phylogenetic analysis. And it looks like that's going to go down to about, say, here. Maybe here's this SVG that everybody has. All right, and so that's actually a good set of sequences to use for a phylogenetic analysis. All right, so we now have, and we go back and we're going to trim to just have a set of sequences where what we're comparing is, if you will, apples with apples. And even though they do have some differences, we feel comfortable we're looking in a region of true homologous sequences. All right, and so in the next video, we'll talk about how we use that set of trim sequences to construct a phylogenetic tree.